The overall goal of the following experiment is to quantify rapid changes in cell matrix adhesion in endothelial cells exposed to matrix-modifying agents in real time using cell-substrate impedance and live cell imaging. In this experiment, the heme enzyme myeloperoxidase is employed as a pathophysiologically relevant matrix-modifying agent. This is achieved by first coating surfaces with fibronectin to provide an adhesive matrix substrate. Myeloperoxidase, or MPO, is then allowed to bind to the fibronectin. Next, suspended endothelial cells are seated onto the surfaces and incubated to attain maximal cell attachment and spreading onto the fibronectin. Then, the cells are exposed to hydrogen peroxide in order to initiate MPO-mediated fibronectin oxidation. The subsequent rapid changes in cell matrix adhesion are then quantified in real time using cell-substrate impedance and live cell imaging analysis. The results show that rapid losses in endothelial cell matrix adhesion triggered by MPO-mediated fibronectin oxidation can be quantified in real time with high temporal resolution and in a label-free manner by cell-substrate impedance and live cell imaging.
Well, the advantage of these techniques is they can quantify changes in cell matrix adhesion continuously with high temporal resolution. And this means that they can provide important new insights into how matrix modifications drive downstream changes in cell morphology and adhesion-dependent signalling processes.
The protocols are of broader significance in that they can be readily adapted.